We've got about 60 people online, and we've got our six experts and presenters. My name is Allan Young. There's a slight change to the format in that I'm going to chair because Samir is going to present. And the paper that this is about is Could History Repeat Itself? The Influenza Pandemic of 1918 to 1919, Schizophrenia and the Immune System. So a few things to just say before we start. We've all become, or many of us have become experts in teams in the last few weeks, from a rather forced expertise. But there's a few things that we should actually go through. One is that if you can blank your screen and mute your microphone unless you're actually speaking. And please, if you can resist the temptation to take control of Samir's presentation or so on and so forth. What we occasionally get is someone who hasn't muted their microphone and we can hear, dogs barking in the background and so on and so forth. Okay, now please remember this is our first virtual meeting. This is actually a historic event, I think, because of this, and it may be the first of many. But bear with us if there are technical difficulties. Okay, we are recording the meeting. Dr. Jauhar will be presenting his slides, and this will be followed by the experts, and then the question and answer session. To ask a question, many of you will know this already, but you go to Show Conversation on the toolbar and write a question in the appropriate box, and then click, and it'll come through, and I will ask it on your behalf. If anyone needs hearing captions for the hearing impaired, select the three dots on the toolbar and press Turn on Live Captions. So without further ado, I'm going to hand over to Dr. Jauhar, who's going to go through the paper, Could History Repeat Itself? I see we've got 119 attendees plus the experts. That's fantastic. Could History Repeat Itself? The Influenza Pandemic of 1918-1919, Schizophrenia and the Immune System. Samir, over to you.
That's super. Thank you, Allan. I think it's worth saying that this came along because we were thinking of the Journal Club, and it was very difficult to see how we could get it going. And we didn't realize the junior doctor brought this up, and then they suggested to me, well, we don't have the time to do this. And I thought, well, I might as well try and help out. So this is my take on the paper. Hannah, can you take me through the slides? And I think can everyone mute their microphones, please, because there's some feedback there. Super. Hannah, so we have and we've also got a mental health blog. So if you Google mental health and that title there, you'll have a blog where I summarize the paper as well, and I can take you through it as we're going along, which I think is probably the most helpful thing to do. Could we have the next slide, please? So again, can I just check, someone's got can everyone mute their microphones, please? Because there is feedback there. Okay, so the background to this was the influenza pandemic of 1918-1919 and trying to link the increased incidence of schizophrenia there to the immune. We've known in psychiatry that pandemics have been associated with symptoms of major mental illness for years. The authors make the point that in 1732, as far back as then, symptoms of hysteria, neurasthenia, melancholia were all noted by people, possibly in relation to delirious states. But fast forwarding, we know more about the immune system in the brain, and the link to psychosis and schizophrenia. So essentially, this paper was an effort to link what we know from the influenza pandemic to what we know about the immune system and schizophrenia. So what was it? It's a narrative review. It covers epidemiology. Sorry, Hannah. Thank you. What do we know? It's a narrative review. It covers epidemiological studies, with evidence for a direct effect, so the effect of the influenza pandemic of 1918-19 on incidence of schizophrenia, as well as talking about the others who were exposed to influenza and subsequent incidents of schizophrenia. It covers ecological studies, case series, population studies, and more specific studies where they've looked at biological markers. The authors then look at the immune system and try and give various mechanisms they think that could be at play these two things together. Could we have the next slide, please? Thank you. So this all really comes along from the way that psychiatry worked in the 1920s. Great descriptions of people having symptoms and people basically within big psychiatric hospitals. We've got a rich history of that in psychiatry, going back to Kraepelin, Bloiler, and the rest of them. Ginger, quite an interesting person, apart from the fact that he lived till he was 96, so good genetics. He was also from a family of psychiatrists, so you could argue probably good genetics there as well, and made a significant contribution to a number of facets of psychiatry, and founded his own psychiatric institute, actually, which is impressive in itself. When he was at Harvard, he was looking after the Boston Psychopathic Hospital. That's an unfortunate name. And he basically noted in a 1926 paper of 100 people who came in having had who came in with acute influenza, 25. Sorry, could someone mute? Could everyone mute?
Samir, so that's the traditional dog barking.
That's dog barking. Sorry, missed the dog barking. So out of the 100 patients who presented acutely with influenza, and they had data on about 80 of them, 25 were defined with dementia precox. It's worth making note of the fact that he distinguished this from delirium, and he distinguished it from other psychoses such as affective psychoses. So out of 100 presentations of people with acute influenza, 25 fulfilled the narrow definition for dementia precox that was given by Kraepelin, I think, in 1908 to 1909. Could we have the next slide, please? And when he followed them up over 1 to 5 years, he found that about 2/3 had a reasonable clinical recovery, which is odd because you'd expect in dementia precox, classical dementia precox, for it not to have a great outcome such as that. And he posed the question, what's going on with these patients? Now, we'll come back to this study, but again, bear in mind, this was 1926, so it was a fair amount of time ago. If you ever get the time to read the paper, it's worth reading for the descriptions. He puts descriptions of a significant number of the patients, and it's beautifully written. These people definitely knew their patients. Could we go to the next slide, please? So that's looking at direct effects of influenza and during a pandemic, exposed to it, and incidence of schizophrenia at that time. The other question that came into people's minds, and Robin will probably talk about this better than anyone else, well, not probably, he will do, was in the early 80s, people noticed that there was a seasonality of birth. So there was an excess incidence of schizophrenia in people who'd had winter pregnancies, and people scratched their heads, probably other parts of their anatomy, trying to figure out, well, what's going on here? That original paper was from rural Ireland with Fuller-Turri's group, and essentially finding an increased incidence of schizophrenia in people who may have been exposed to some measure in winter. This led to population in the 80s and 90s, with case register studies in Finland and Scandinavia, where they've got good data sets, finding an increased risk. This was meta-analyzed in the American Journal in 2010, and the authors found an odds ratio of about 3, which for anyone in epidemiology is reasonable. So again, you have exposure to influenza during pregnancy and an increased incidence of schizophrenia. Again, what are the weaknesses of that literature? It's self-report. It's not a biological measure. You've got conflicting findings from different studies, and you have methodological problems. And the authors make that point. Could we get to the next slide, please? So looking for a better measure of whether someone's been exposed to influenza or not would make sense to look at biological measures. So you'd look at serology and see whether there's evidence biologically of someone being exposed to influenza. Great idea, very smart idea. Unfortunately, not very many studies, as the authors point out. 2 meta-analyses done. I'll focus on the second one, which is the one looking at prenatal exposure. So at prenatal exposure to influenza, where you find the serology, 2 studies in the literature with an increased risk, up to a 3-fold risk, not powered enough to have a statistically significant difference, and you wouldn't bet your mortgage on that. In terms of childhood exposure, so again, I guess you're just looking at it as an exposure variable. There's some evidence to suggest viral as opposed to bacterial infections increase risk of schizophrenia, and the authors found a risk ratio of 1.7. That author is Golem Kundakar, who's gone on from there to do a lot of sort of studies with depression, schizophrenia, and the immune response. Could we have the next slide, please? So, looking through that and trying to make sense of it, well, you have the neurodevelopmental hypothesis of schizophrenia, an exposure to something whilst in development affecting development itself. I think if you're going to look at the prenatal exposure, you're going to have the issue of the fact that someone's had prenatal exposure, they'll have increased obstetric complications, and that's an independent risk factor for psychosis. Robin will talk more about me, but what's interesting is he wrote this in the Lancet whilst it was raining in Italy, apparently one afternoon. Halcyon days. Next slide, please. Thank you. So, having put forward the epidemiological evidence, the authors then go on to say, well, what are the causal mechanisms? What's going on with the immune system, and can we link them together to schizophrenia? So the first thing they take is maternal immune activation, and they use models, so animal models, more specifically rodents, and exposure to influenza or a pathogen in utero, and seeing the effects on what we would consider markers of schizophrenia. The markers would be gene and protein expression, neurotransmitters. The neurotransmitters that they link are serotonin and taurine, and prepulse inhibition. So prepulse inhibition being decreased in schizophrenia, where someone gets a weak stimulus and you get a startled response. And that's been a replicated behavioral finding in schizophrenia. They conceptualize that's mediated by a cytokine response. Cytokines being the signaling proteins that are implicated in the immune response for anything, essentially. So they're the proteins that are secreted whenever you have an immune response. Immune response being, at a simple level, our response to, well, inflammation being our response to insult or injury. Could we press the button again? Next slide. So that was the first way that the authors try and make sense of it, is maternal immune activation using animal models for what we might consider some markers of schizophrenia. We'll come back onto that in regard to the strength of that finding. And then the authors talk about autoimmunity. So what's autoimmunity? You'll all know that that's when our immune system targets its own cells, and we basically have our own immune system targeting the host cell. They've used examples of autoimmune encephalitis. So that's when, about 10 years ago, or probably longer, Tom will talk better about that, where we found antibodies to receptors in various parts of the brain associated with psychosis. They found the NMDA receptor antibodies in people with psychotic symptoms, and not just people with delirium psychotic symptoms or encephalitis, so an infection. And the authors also talk about molecular mimicry. So they talk about a pathway where you could have cellular expression linking exposure to a pathogen and then the host cell having an autoimmune response. So basically trying to find a way to link exposure to a pathogen and the host cell doing damage to itself. using the example of NMDA receptor antibodies, because that's something that's been looked at for the last 10 years and been associated with psychotic illness. Could we have the next slide, please? So strengths and limitations of the paper. I've done a peer here, so just press the button, it'll come through, please. Thank you. So first off, a limitation is it's a narrative review, which is something we all like, but it's not systematic because there's not enough studies to do a systematic review. And I think that's entirely fair. What I liked about the paper is it's suitably cautious. It's not saying there's a definitive link. And when they've gone through the epidemiological evidence, they've presented it as it is. They haven't just presented what seems to suit them, which is what I like. In terms of the epidemiological evidence, I've put down the Redua paper there because I think that's the best paper we have in regard to causes of psychosis. It's an umbrella review which looked at a lot of studies, and it looked at all the systematic reviews and meta-analyses and reanalyzed them to find causal factors for psychosis. So a limitation of the study possibly is that influenza doesn't come up in that as a risk factor. The reason for that, well, the systematic review covering influenza there only covered a few studies with very small sample sizes. So you can't really say that detracts from what the authors try to say. So that's the epidemiology covered. And the epidemiology, I think it's reasonable to say, is a little bit murky. So in regard to the immune work, well, it's animal models of psychosis and psychiatric illness. This is something I'm always reminded of when you think about animal models. You know, have you ever met a hallucinating mouse? You might well have done, ladies and gentlemen, but you probably wouldn't know that they were hallucinating. And the other issue, I think, is with the NMDA receptor antibody hypothesis of schizophrenia, because Tom will update you on this, but there are a few studies where healthy controls have also had NMDA receptor antibodies, and so it's not as definitive as we might like to think. Could we have the next slide, please? So when I was thinking about causality and presenting anything in terms of causality, I guess you have the Bradford-Hill criteria, which was used for examining smoking and lung cancer. That was Austin Bradford-Hill and Richard Dahl, where it was very clear there was an association between smoking and lung cancer when you use these criteria. So how well do they work for influenza and schizophrenia? Well, strength of the association, there's some strength there. Risk factors, odds ratio 1, 2, well, going up to 2 or 3, that's okay. Consistency, not enough studies to be able to say that. Specificity, can any viral infection be associated? Possibly. Temporality is covered in terms of direct effects and the effects on offspring. Biological gradients, is there a dose response? I think it's difficult to measure that in terms of severity on the evidence we have. Is it plausible? Well, I think it's a beautifully written paper, right? It's plausible to me, but that could just be covering my inconsistencies. Is it a coherent hypothesis? I think it is. Are there experiments that have been done which help us with regard to that? Yes, but not as many as we'd like. Could we have the next slide, please? So in summary, in regard to causality, I think there's a reasonable case there that we can cover that with the experts. So let's be honest here. The reason we're covering this now isn't because anyone's scared about influenza. We've got a vaccine for influenza. The thing that's changed our lives, I think, beyond any doubt, is COVID-19. So what are the similarities between COVID-19 and influenza? Well, they're both respiratory infections, though. Hannah, could you press the button, please? I think it's worth reiterating that it's not the flu, despite what this great scientist here thinks. And they are different entities. Could we have the next slide, please? And I think it's worth leaving you with this paper, which is quite nice, and Allan will cover some other things. But there does appear to be involvement of the central nervous system with COVID-19. So this is a retrospective study of 214 cases in Wuhan, China, where we all know COVID had its genesis, or COVID-19 had its genesis. They found about a third of people had neurological symptoms. And when you look at the neuropsychiatric symptoms, and you could question and you could think about the various etiologies happening there, the things that we've discussed, whether it's effects of fever whether it's effects of small little cerebrovascular accidents. It seems to be associated in COVID with more severe illness and respiratory illness. And it'll be interesting to see really what comes out in terms of the literature in regard to exposure to COVID and manifestations in the central nervous system. And lastly, there, offspring and risk. I think life's always more nuanced, as is science, especially schizophrenia research, than any of us would ever have thought a number of years ago. I like to think we're thinking about possible increased risk and possible, not, possible is the operative word here. Thinking of things as a multi-hit hypothesis. That's Keshovan's hypothesis, I think, from 1999, where you get exposure to a number of cumulative risk factors before developing what we might consider the construct of an illness. So I think that's my main take on things. I've hopefully presented the epidemiology, or what little epidemiology there is, but it's rich, of what the authors have presented. I think they'll cover the immune response there. But for me, it was a nice paper. They could not possibly have imagined when they wrote it the relevance it would have to just now. But I'd like to thank the authors for doing it, and I'll leave you with people much more intelligent than myself to muse on all the findings. Thank you.
Thank you, Samir. I think at this point we usually allow people to ask direct questions of the presenter, just about any points of clarity on the paper. So just to remind the audience, we've got over 180 people on the call if we include the experts, which is really quite impressive. So I'm just looking to see whether there's someone saying nice presentation. I must say that you really, I think, caused a real change in my view of the world, Samir. I'll never see another mouse without being able to wonder if it's actively hallucinating. That's perhaps just me. So if there's no questions if there is questions, I'm not getting them, but I can't see any questions. Okay, let's just check. Nope. Okay, well, let's move on to
There's one question from Marta DeForti.
Oh, is there? No, I can't see those, Charles. Do you want to read the question out then?
Sure. So could we have more details of the neuropsychiatric symptoms associated with COVID-19? Great presentation, Marta.
Right. Thanks, Marta.
Yes, this is the JAMA neurology paper, isn't it? So fluctuant consciousness, they found hard neurological symptoms as well. What's probably best is if I find the link for it and just go through it, because it's 214 folk, a third. It was a smattering of really everything, to be honest. They didn't do in-depth psychiatry or in-depth psychiatric symptoms, but clouding of consciousness, symptoms of delirium, neurological symptoms as well, so hard neurological symptoms as well. I would have to check the rest of it and check the rest of the paper, though I'm sure with this amazing technology, we should be able to put, if I send Hannah the link to the paper, Hannah, could you put that on Teams? Is that all right?
Yeah, or we can send it round afterwards.
Perfect.
Great. In the spirit of making it up as we go along, I'm going to suggest that Charles alert me to any questions, because I did get Marta's question, but it was only halfway through Samir's answer. So there's obviously a delay for some reason with me. Okay, we'll go on to the experts now, and I'd like to start off with, I think they're all on the call, Miss Adrianna Kepinska. Adrianna, do you want to just give us your view of this paper?
Oh gosh. Well, thank you so much, Samir, for a lovely presentation on the paper and for the blog as well. I'm gonna put the link to the blog in the comments, where Samir also gave really nice comments on the paper. I mean, I think I think the Samir's main point on our paper was that it was not a systematic review, which I definitely agree with, and I think it would be really interesting to kind of look at this content in the sort of more systematic way. That's sort of like a follow-up question that I will have. If we know of many papers that are systematic reviews of COVID related research?
Mm-hmm. Well, I mean, I think that's a great question, because I think there's going to be lots of systematic reviews done, given that people can't do much face-to-face research. So
Sure.
I suspect the first thing to do would be to see what's been registered in Prospero in terms of current reviews. Okay. Anything else from you, Adrianna?
No, not really. I think Samir did a great job.
Okay, good. Rajeev, Krishnadas, are you on the call? Rajeev?
Hi there.
Hi.
I'm here. I'm from Glasgow. Hi.
Oh, great. You're calling in from Chicago.
No, from Glasgow. Sorry.
Oh,
Not that
Glasgow.
far, yeah. Just
a much more foreign place than Chicago. Do go on. Tell us your considered views on this.
It's a very good paper, and I think it's a very good review of the whole thing. My own background as my PhD was looking at how peripheral inflammation affects serotonin transporters in the brain, and also looking at other correlations between circulating inflammatory markers and cortical thickness. And one thing, what I thought was, yes, and I was...
Rajeev, could you speak up a bit? I can't quite hear you, and I'm sure others might have trouble as well. I don't know if you could move closer to the microphone. Yeah.
Can you hear me now?
That's a bit better. Thank you.
Okay, yeah. So yeah, my was sorry. Did I do something wrong?
No, our sincere apologies. It actually came up on my screen who the culprit of that is, but I'm not going to out them to the entire 180 people on the round. But they know who they are. I think that's a ringtone. Sorry, go on, Rajeev.
Okay, you can hear me. Yeah, my PhD, I looked at how circulating inflammatory markers are associated with serotonin transporters and cortical thickness in healthy people and in people with arthritis, so in chronic inflammatory conditions, basically. And interestingly, we've also just finished a study looking at typhoid vaccination affects the connectivity within insula and other cortical regions. And we find that, inflammatory, there is actually quite a lot of association between inflammation and various, whatever phenotype you look at in the brain, there is some association. And I would be very surprised if this didn't have an effect in vitro, as in pregnancy as well, especially when neuronal migration happens during the first 2 or 3, the second trimester basically, and especially the GABAergic neurons. When they migrate, that's very much regulated by chemokines, which are basically cytokines. And if I would be very surprised if if you disrupt the maternal inflammation, that that wouldn't affect the GABAergic migration. And we all know that GABAergic dysfunction is now quite crucial, as in the excitatory inhibition imbalance is quite crucial in patients with psychosis. So all that is quite. I think I'm not very surprised by this. But what reading this, something which came up in my mind, and I don't want to be controversial, and I think it is a controversial subject, given all the Andrew Wakefield stuff. Given an infant or a mother is also exposed to a number of immunization, will that have an effect? This is just a query. This is a complete query, and probably Tom can kind of answer. Given the infant immunization schedule, where over the first 3 months, I don't know, they get immunized with quite a lot of stuff. Will that have an effect on any of these with neuronal pruning, or it's something which it came to my mind when I read this paper. That's it.
Okay. Well, thank you very much, Rajeev. Can I just say that there's a very nice suggestion from Adrianna that if you put the live captions on, you can hear you can read what people are saying. And someone was typing there, so could I just get you all to check that you've muted? But if I can very broadly summarize what I think Rajeev was saying, he was saying there is evidence of inflammatory changes in the brain, and these may be associated with long-term sequelae. Is that correct, Rajeev?
Sorry, could you repeat that again? Sorry, I couldn't hear you.
Yeah, I was basically trying to summarize what you were saying is that there may be reason to believe that there are changes in the brain, and probably inflammatory changes, and these may give rise to long-term sequelae.
Yes, absolutely. That's exactly what I was trying to say. Yes.
Okay, let's move on to, thank you very much, Rajeev, and thank you for calling in from Glasgow. And let's go on to Tony Vernon. Anthony Vernon, are you on the call, Anthony?
Hi, good afternoon, everybody. Can everybody hear me well?
Yes, we can hear you, thanks.
Perfect. So I just want to start by saying thanks very much to Samir for organising this, and also Hannah for making sure all the technical aspects are working. I wanted to speak specifically a bit about the animal models, because I noticed Samir mentioned that, and particularly hallucinating mice. And I think I need to say a few points on this. I'll try to be brief. I think the first thing to say is the importance of these animal models is to say, you know, they're just there to test a hypothesis that if you expose the mother to an infectious pathogen, do you find any evidence for kind of vertical transmission of that pathogen? Or is it that the associated immune response in the mother can cause some behavioral disruption later in life or some effect on brain development? And if you look at most of the animal studies summarized in the paper, then that's exactly what they're testing. They're looking at whether influenza infection triggers any kind of pathology and so on. And I think summarizing that, what you can see is that there's little evidence for this kind of vertical transmission idea, and putting that into the current context of COVID, that also seems to be the case, or I appreciate there's very limited evidence. And so it is something about this maternal immune response that's important. And then I think the second important thing to say then about these models is that they are just models of maternal immune activation. I think something that the animal field has always suffered from, and we've got better at correcting now, is to say, well, it's a model of X or Y or Z, when clearly, you know, we cannot do this. We're just modeling one risk factor in a sea of genetic and environmental factors. And the important aspect there is also that the MIA is in no way specific. So you can find associations for autism, for depression, and so on and so forth. So, you know, think of it more as a priming model, perhaps. And the idea then is useful because these animals have you know, been helpful because they've given causality or a nexus of causality to the epidemiological data, and then done something that, you know, has been lacking, which is to try and speak to mechanisms. And we've already had a couple of mentions there about GABA interneurons. There's very, very good evidence now that the migration of these neurons is affected in these models. There's good evidence for dopaminergic deficits that we all know and love. And there's also good evidence for lots and lots of epigenetic changes that occur very early and affect how cells are developing, particularly microglia and so on, which are critical in participating in how the brain is wired up. And so, you know, there are a lot of strengths there that we should consider. On the flip side, if we look at them objectively, we can also find a number of limitations. And the sort of first of those is the nature of the stimulus. So, you know, obviously models where they use an actual viral infection may be argued to have greater relevance. People started to move away from that and use synthetic toll-like receptor 3 ligands because that's simply a biosafety issue. You don't need the same level of containment to do those experiments. And what people have found using those models is that an acute stimulation with poly-IC, for example, triggers a very massive maternal immune response. You get to see tremendous increases in cytokines, particularly IL-6 and IL-17. But if you sort of look at the fold change of those cytokines, and compare that back to data from blood spots, for example, in maternal circulating cytokines of their children actually then were later diagnosed with say autism, and there was a link to infection, you can see that the concentrations are very, very different. And so perhaps a big criticism is the physiological relevance of these models. What are we really triggering there? And would it be better to look at a more low-grade chronic immune response that hasn't really been done? And so the other, of course, criticism is that they're animals, and so what relevance do they have to human data? And I think that's a fair point. We need to think very hard about the endpoints that we look at when we study these animal models. Are we looking at behavioral markers that have dimensional relevance in terms of, say, cognition or social behavior? I mean, but we can't interpret those too far. There's also questions about species differences. Human brain development is much more complicated. There's a lot of key molecular differences, and that's not something that's really been explored. So I think there's a need for complementary systems, using induced pluripotent stem cells and so on, to try and understand what are the effects of early stimulation with different cytokines on how these cells develop and connect and so on, accepting that that's also a highly artificial system, so they're complementary rather than replacing each other. And with Deepak Srivastava, we actually have there's a preprint now led by Deepak on bioRxiv on the effects of interferon gamma on neuronal development, which you might find interesting. And the argument is then, well, how do these data translate to humans? And actually, there is quite some growing evidence that the data from the animal models do have relevance, particularly the work from Claudia Buss and Damien Fair, which show that maternal circulating IL-6 is actually directly correlated to connectivity of the amygdala in the offspring. And that's also related to externalizing behaviors in those children at age 2. You can also predict maternal serum IL-6 from neonatal brain networks. So clearly there's a connection between how the brain is wiring and functioning and the degree of which immune exposure is happening. So I think that's starting to say, okay, there is something important here. One last point to mention is something that nobody spoke about so far, which is, the odds ratios are there, but they are very modest. And, how much of a concern should this be? And the idea of risk and resilience is something that we need to think about. So, not everyone who's exposed to such an infection would have any kind of adverse outcome. And what are the biological mechanisms that are driving risk and resilience? And I think that's really something that's been lacking in the field. Working with others in the MIA model world, we're trying to address that question now. And hopefully we'll have an update for the community soon. But I think, we do also need to think about maybe other intermediate models. There's now a very good primate model of MIA, and that brings logistical challenges, of course, but there's also now a primate model for COVID-19 infection, just published in Science. So I think, merging some of these ideas offers a lot of good opportunities. But I'd be happy to answer any other comments or questions about the animal work.
Yeah, thanks very much, Tony. And I mean, I guess there's a couple of points. You've very expertly outlined the strengths and weaknesses of the animal work, or the animal models rather. But I suppose one point that's particularly important to me is that the neuropsychiatric or neuro and psychiatric sequelae of COVID-19 infection may take years or decades to fully unfurl, and therefore, I guess clues from rodents might be particularly helpful because you can get those in a matter of weeks or months rather than years or decades. I mean, I guess that's part of the point that you're making.
Yes, absolutely, and I think we could already start to look at that. we know, for example, IL-6 again seems to be important in the sort of in some cases where we've read about cytokine storms in these patients. And animal studies have shown very nicely that if you give influenza or poly-IC to an animal that lacks IL-6, then you don't get a phenotype. So, clearly specific cytokines do have an effect, and that's something we could immediately begin to look at.
Yeah, sure, absolutely. Yeah, I'm struck with the parallel with the hunger winter and the Chinese famine, where really it took a very prolonged period of time for the epidemiology to bear fruit. Okay, let's move on to our next expert, Dr. Tom Pollak. Tom, are you online? Are you able to comment?
Yeah, I'm here. Yeah, thank you, Samir, for that summary. That was excellent. I just want to provide, I guess, just more of a kind of clinical spin on things at first before coming onto the science for a little bit, but just to emphasize quite how sort of strange it must have been to be a psychiatrist working in 1918 or 1919. And it really sounds like there was a tidal wave of neuropsychiatric cases affecting almost anyone who had been working in psychiatric practice at that time. The Menninger paper, which, although Samir says it's from 1926, there was an earlier one from 1919, so written pretty much contemporaneously with the Spanish flu outbreak. It describes a period of 3 months in a single hospital when 100 patients came in with acute influenza-associated psychoses. And that's really quite incredible if you sort of compare it to what's happening now. We're clearly not seeing anything like that degree of presentations. Not only that, but of course, about a year earlier, possibly a little bit earlier than the Spanish flu, but we're not really sure, there was another epidemic going around the world, sweeping the world, and this was the the epidemic of encephalitis lethargica, which is this other severe neuropsychiatric disorder which had very bizarre presentations. People fell asleep, they became catatonic, they had all kinds of sort of neurological symptoms. They would develop acute mania alternating with sort of somnolence. And again, one sort of very influential theory of that suggests that may have been a post-infective process, some sort of immune response to influenza infection. And if you read some of the small print of Menninger's series, there's some incredible stories. He describes 3 patients at one point who, in the previous epidemic flu, which was I think in the 1890s, this was I think the Russian flu, they presented with a first episode of psychosis. They then got better, and then in the Spanish flu of 1918, they had a second episode. And so there's clearly, for at least some of these patients, there was something specific about the influenza. And the problem is that we don't know what the mechanism was, whether it was direct infiltration of the brain, almost certainly not, whether it was a sort of post-infectious or para-infectious kind of immune response, or even an autoimmune response. There's some suggestion that it could have been an autoimmune response in some of these cases. But to just sort of bring it up to date a little bit, I think Marta was asking about what do we know about the current presentations of coronavirus. Is it possible that we'll see anything like what we saw 100 years ago? And I mean, Samir makes reference to one paper, but actually there have been 25 papers so far published, either sort of formally published or in preprint, outlining neurological or neuropsychiatric manifestations of coronavirus. And they all kind of lean heavily on the very neurological side. So they're, at the worst sort of instance, there are cases of acute necrotizing hemorrhagic encephalitis. And on the sort of the milder side, there's encephalopathy, which probably is a bit of a kind of posh word for delirium. But one of the things I've been struck by is speaking to colleagues, and some of the cases that I've heard anecdotally, cases of catatonia being associated with coronavirus infection, and speaking to some of my colleagues in old age psychiatry, who a number of separate colleagues have said something very interesting, that they are seeing a lot of delirium, but that delirium is not the sort of typical delirium that you might see in an older person. It's not characterized by really severe or acute confusion. Sometimes the confusion is relatively minimal, but the psychotic symptoms appear to be a lot more marked, particularly the delusional symptoms. And actually, if you look in the Menninger paper, this gets commented on, a very similar sort of presentation, to the extent that he even refers to it at one point as delirium schizophrenoides. And so, who knows whether we're seeing something that, at least in small part, might resemble what we saw then. There is an answer to that, and I think actually Jenny Drive posted it earlier, but any of the psychiatrists here will have received an email from Wendy Byrne yesterday. I've just sent the link again. And so some of us in the neuropsychiatry faculty, in association with people from the Association of British Neurologists, we've set up a case monitoring system whereby psychiatrists now can register any case of a patient that they see who has a sort of interesting neuropsychiatric or psychiatric or even neurological presentation in association with suspected or confirmed coronavirus infection. And we would really encourage as many people as possible to do that. And it doesn't matter if it turns out that that association is purely coincidental, or in fact it turns out they didn't have coronavirus. But the urgency of this is that we do need information, and we think this is going to be one of the best ways to do it. So please do report if you can. And I can just say a little bit about, I think somebody asked a question about an increase in the number of people presenting with the first episode of psychosis. I don't think there's any formal data about that currently. And I mean, certainly we're seeing in liaison psychiatry quite a lot of first episode mania or psychosis occurring in people who just aren't really the right kind of age. So people in their 30s, 40s, 50s, and 60s, which we think is a little bit unusual, but none of these people are testing positive for coronavirus. So we can't really discount the enormous influence of just the sort of the paranoia and the stress of living through a pandemic. So we might be barking up the wrong tree when we're thinking purely about these biological stories. I think there's one other relevant piece of information, is that in 2010. A group led by Bob Yolkin from Johns Hopkins University actually measured antibodies to coronaviruses in patients presenting with a first episode of psychosis. Now, of course, this wasn't the coronavirus that we know now, neither was it MERS or SARS. It was the four main coronaviruses that cause seasonal sort of coryzal, coughs and colds. And they did show that the levels of antibodies, or the levels of immunoreactivity to those coronaviruses, or two of them at least, were increased in patients with a first episode of psychosis compared to healthy controls. And although that's by no means definitive and doesn't really suggest anything for sure, I think what it does is it represents another kind of methodology that we can be thinking about now. So rather than just looking at these patients who are in ITU or who are presenting with acute respiratory symptoms and seeing how many of them become psychotic, well, we know already that probably a fair old proportion of coronavirus infections are going to be either asymptomatic or very mild. So maybe what we should be doing is looking at people who are presenting with a first episode of psychosis, and then trying to find serological evidence of their antibody response. Are they showing a clear immunoreactivity to coronavirus, and whether that differs from either healthy controls or from other psychiatric groups. And I think that way you actually get a far more representative kind of picture of what's going on by not just focusing on these severe cases who are obviously going to be more likely to develop mental illness, not just because of the fact of the infection, but because they've been on ITU, they've been isolated, et cetera, et cetera.
Okay, well, thanks very much, Tom. And of course, I think the first episode of psychosis is going to be something that will require time to evaluate. Samir's asking if we should test all new cases of first episode psychosis for COVID. I would have thought that would be perfectly reasonable to do. There is an emailed question from Simon Wesley about the increase in Parkinson's after the epidemic, the pandemic that was in Samir's paper. Samir and Tom, do you have any comment on the Parkinson's increase?
Yeah, I think Simon picked up on that very clearly, as Tom picked up on encephalitis lethargica. What some people, I guess, aren't aware of is with schizophrenia, we've known for, I think, since about 1920, that people do present with dyskinesias before even being offered neuroleptics. So there is something happening with the dopamine system, and it is in disequilibrium, essentially, before people are actually offered neuroleptics. So there is something happening with that system. And I think that's a vital piece of information. And if you think, yes, the Parkinson's stuff you couldn't miss, what Tom picked up beautifully there was that also the clinical descriptions, you never know how good they were of a proportion of people with Parkinson's or the people with bradykinesia, with encephalitis lethargica. And my suspicion is there are a lot of sort of psychotic symptoms probably going on in people. But essentially, great point by Simon. But I think what's interesting is people before, people with schizophrenia developing movement disorder before being given the medicines, which I think is fascinating.
Yeah, my recollection is that Robin McCready did a couple of nice papers from India with people in India who had schizophrenia who'd never had antipsychotics, and not only did they have a significant amount of dyskinesias and so on, but these would also fluctuate during the course of the illness, because he did a follow-up paper as well. Now, Ranga has a point about what serological tests we would need to do for first episode psychosis. I guess that would actually, Tom, can you answer that briefly, Tom Pollak?
Yeah, I think at the moment the problem is we don't have those serological tests widely available. They're only happening in a research capacity at certain labs. So I know Johns Hopkins, amongst others, have been leading on this. I think the problem is with all this serology is and this is a problem that the government are facing at the moment, is we don't know what to make of a positive test, we don't know how to threshold the test, and we don't know whether the test provides evidence of lasting immunity at all. So I think, for now, I think that of course there may be an argument for testing for acute COVID infection in our patients. I think that's important, but I think that the sort of testing to kind of demonstrate a kind of causal relationship between asymptomatic infection and psychosis, I don't think it's quite ready for clinical practice at the moment.
I can perhaps add to that, because we've in the Centre for Innovative Therapeutics with Mitchell and Steve Williams, Eric, myself, and others, we've been working on a serology test with Jonathan Edgeworth from Guy's, and there's a test which is currently stuck in Chinese customs, but it's passed with a suitable fit for purpose sensitivity and specificity, and we've got 7,000 of those tests which we're trying to get in the next couple of weeks for the Denmark Hill campus. So again, it's the questions about what this test tells us for long term about immunity, but certainly tell us if people had it, and so far it's been the best test in terms of actual sensitivity and specificity. So there's a glimmer of light with regard to that. Okay, we've got about 5 minutes left. Let's go to Robin Murray for summing up. Robin.
Well, I was going to add a few historical points, and we've heard about Menninger, but the other important figure in the influenza story is Sarnoff-Mednick, and he was the man who had the idea to look at the effect of the 1959 Asian flu epidemic. And I remember hearing him report that study at a conference and thinking it was a mad study. So we went home and set out to disprove it. And to our astonishment, we replicated the effect. And then there was a whole range of studies looking at, did exposure to influenza during pregnancy increase the risk of schizophrenia? And one of the problems was, of course, what is a replication? The original theory was exposure in the 5th month, but some people got exposure in the 4th month or the 8th month or the 3rd month, and they said this was a replication. So it was very difficult to know whether this was true or not. You might say that the serological studies may be much more valuable, but there were serological studies where they found serology from pregnant ladies at the time of the influenza, and they were able to look at the teters, maternal teters versus the occurrence of schizophrenia. These are all from the same one group, and it's a group who tend to get very positive studies. So I think one has to be a little skeptical. We actually tried to do one of the first studies of looking at an animal model. So we, with a chap called David Cotter, we got little mice, and we were about to make these, give influenza to these pregnant mice. One thing nobody's mentioned is that different epidemics have very different effects. And so if you study, say, the 1968 influenza, and you get no effect on schizophrenia or no effect on the animal model, that doesn't mean to say that the 1918 or the 1959 epidemic strain would not have had an effect. So we decided we'd use the 1959 strain, and we went off to see an expert called John Oxford and explained the story to him. And he said, "Oh, you're going to become very famous. Perhaps actually infamous. How old are your technicians?" And we said, "What's that got to do with it?" And he said, "Well, if your technicians were not born and exposed to the 1959 influenza virus, you'll probably start another pandemic." So at this point, we recoiled and decided just to use a mundane, more recent version of the virus, and it did show that the offspring of the pregnant mice did indeed have, rats did indeed have decreased hippocampal volume. So I'm much more interested in the maternal exposure hypothesis. I'm not very convinced of the effect of exposure to influenza as a precipitant of illness. Of course, there is some suggestion it can precipitate NMDA encephalitis. Some of you will remember the famous case of the German brown bear in Berlin Zoo, who died of encephalitis, and this was due to him having caught influenza from his keeper 2 or 3 weeks before. But I think it's much more interesting to look at the effect during pregnancy, and that could be a very general effect. It could be that that people who are exposed to influenza, the mothers who are exposed, they're more likely to be generally sick, to have poor nutrition, to take all sorts of medication, even some whiskey to try and get rid of the symptoms. They're more likely to be premature. The babies are more likely to be premature. So it could be a general effect, but cytokines, a cytokine reaction is an interesting possibility. And Tony mentioned that COVID, that IL-6 comes up in some of these animal models. As I understand it, a massive increase in IL-6 is the best predictor of a bad outcome in COVID. So I think it may well be that we should look more carefully for the effect of COVID on pregnant mothers, rather than look at first episode psychosis.
Great. Well, thank you very much. We're almost out of time. I'm just looking through the chat just to see if there's anything that's particular that we need to address. There's a few questions which look as though they probably need followed up, so I can urge you all to get in touch with the presenters. I'd just like to thank all of our experts for giving such well-considered and expert input. And I think it was really an exemplary performance by all of you into a topical subject. I'd like to thank Samir, who's done such a great job, really. And I think this should really be the first of many of the remote grant rounds. I'd like to thank Charles and Hannah for doing such a great job in making it happen. And I'd just like to end in the last 30 seconds with one reflection. That if you look at the history of the model, that back when it first came into being about 100 years ago, and there was so much concern about shell shock and military psychiatry, that at that time there was a real concentration on both what we would now call psychological medicine as well as neuropathology. And those two strands still seem to be very evident in the way that we treat this class of diseases at the moment. So on that note, I'd like to thank you all for coming. We've had a great turnout, and hopefully we'll be able to do this again. Thank you very much.